Okay, sir.
Okay. So the screen sharing is disabled for the timing.
Yes. Can I help you to share your screen your, by yourself?
Uh, yes, I'm trying to share the screen just to check if everything is perfect or not, but it's showing that the host has disabled
Okay.
screen.
Okay, sir. Good evening, Mr. Tarun. How are you?
Hi, good morning. I'm good, fine. Thank you.
So do you want to share your screen later for your presentation?
Yeah, I just want to check. Yeah, but it's for the timing, it's showing that.
Oh, okay,
Uh,
okay. Okay. Okay, Mr. Tarun, we're gonna put you to be a co-host in a couple of minutes, and maybe in a few minutes we will start the event.
Iya. Stikes Alifah Padang adalah salah satu perguruan tinggi kesehatan yang ada di Sumatera Barat, memberikan kontribusi terhadap pendidikan kesehatan di Sumatera Barat. Stikes Alifah Padang telah terakreditasi oleh BAN PT dan seluruh program studi di Stikes Alifah Padang telah terakreditasi oleh LAM PTKes. Stikes Alifah Padang mempunyai visi menjadi institusi kesehatan yang unggul dan berdaya saing nasional. Salam Indonesia Sehat, salam Indonesia Maju, Stikes Alifah Jaya.
kepada Stikes agar berupaya keras dalam peningkatan mutu lulusan sehingga lulusan dapat bekerja di tengah-tengah masyarakat. Kedepan, yayasan akan selalu meningkatkan penambahan program studi sesuai dengan yang dibutuhkan oleh pemerintah dan masyarakat.
Stikes Alifah Padang siap berkontribusi pada pembangunan kesehatan di Indonesia, khususnya di Sumatera Barat. Mari bergabung bersama Stikes Alifah Padang.
Yayasan selalu mendukung semua program-program yang sudah direncanakan oleh akademik, yaitu Stikes Alifah Padang, dan selalu memotivasi dan memberikan support setiap kegiatan Stikes agar Stikes Alifah bisa lebih maju, berkualitas, dan profesional.
Stikes Alifah Padang adalah salah satu perguruan tinggi kesehatan yang ada di Sumatera Barat, memberikan kontribusi terhadap pendidikan kesehatan di Sumatera Barat. Stikes Alifah Padang telah terakreditasi oleh BAN PT dan seluruh program studi di Stikes Alifah Padang telah terakreditasi oleh LAM PTKes. Stikes Alifah Padang mempunyai visi menjadi institusi kesehatan yang unggul dan berdaya saing nasional. Salam Indonesia Sehat, salam Indonesia Maju, Stikes Alifah Jaya.
kepada Stikes agar berupaya keras dalam peningkatan mutu lulusan sehingga lulusan dapat bekerja di tengah-tengah masyarakat. Kedepan, yayasan akan selalu meningkatkan penambahan program studi sesuai dengan yang dibutuhkan oleh pemerintah dan masyarakat.
Stikes Alifah Padang siap berkontribusi pada pembangunan kesehatan di Indonesia, khususnya di Sumatera Barat. Mari bergabung bersama Stikes Alifah Padang.
Good morning, ladies and gentlemen. Before we start, I would like to read the rules for today's webinar. Please listen. Welcome to the first Alifah Health Science College Symposium. Webinar Code of Conduct, first Alifah Health Science College Symposium. Number 1. On behalf of first Alifah Health Science Symposium, we sincerely apologize to participants who cannot join the Zoom due to the limited quota. Number 2. For the participant who cannot join the Zoom, you can join via YouTube live streaming at Stikes Alifah Official. Number 3, the participant must input their name and institution in Zoom and YouTube chat room by following the format, name and institution. Number 4, the microphone must be on mute. Number 5, the participant must fill out the attendance link that will be shared at the end of webinar via Zoom and YouTube chat room. Number 6, the participant can ask a question through the link that will be provided in Zoom and YouTube chat room. Number 7, the participant have the some right in participating the symposium via Zoom and YouTube based on the conditions set by committee. Number 8, the participant who have no pay the administration fee will be given certificate SKP by PPNI, EB, and IAKMI. Number 9, an error in filling out the attendance link will result on error in writing the e-certificate. And the last, a certificate will be distributed at 7 days after the symposium. Thank you. Selamat pagi, partisipan yang berbahagia. Sebelum kita memulai acara ini, izinkan kami di sini membacakan tata tertib untuk acara simposium. Yang pertama, atas nama First Alifah Health Science College Symposium, kami mohon maaf yang sebesar-besarnya kepada peserta yang tidak dapat mengikuti melalui Zoom karena keterbatasan kuota. Kedua, bagi peserta yang tidak dapat mengikuti Zoom, dapat bergabung melalui live streaming YouTube @Stikes Alifah Official. Yang ketiga, peserta wajib memasukkan nama dan institusinya di chat room Zoom and YouTube dengan mengikuti format nama dan institusinya. Yang keempat, mikrofon harus dalam keadaan bisu. Yang kelima, peserta wajib mengisi link absensi yang akan dibagikan di akhir webinar melalui Zoom dan chat room YouTube. Selanjutnya, peserta dapat mengajukan pertanyaan melalui link yang akan disediakan di Zoom dan chat room YouTube. Selanjutnya, para peserta memiliki hak yang sama untuk mengikuti simposium melalui Zoom dan YouTube sesuai dengan ketentuan yang telah ditetapkan oleh panitia. Selanjutnya, peserta yang telah membayar biaya administrasi akan diberikan sertifikat SKP oleh PPNI, IB, dan IAKMI. Kesalahan pengisian link absensi akan mengakibatkan kesalahan penulisan e-sertifikat. Dan yang terakhir, e-sertifikat akan dibagikan paling lambat 7 hari setelah kegiatan ini. Terima kasih. Izin Bu As untuk memulai acara, Bu. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Peace be upon of all us. Good morning, ladies and gentlemen. Welcome and thank you for the all attendees who have joined us in Zoom or YouTube live streaming Stikes Alifah Official for international webinar First Alifah Health Science College Symposium, with topic Emerging and Re-emerging Infectious Disease, Future Challenges and Strategy. Ladies and gentlemen, here I am Fatmi Nirmalasari, as the Master of Ceremony will guide this webinar. Before starting our agenda, let me explain the agenda schedule of this webinar. The first agenda is opening. The second is listen to Asma'ul Husna. The third is listen to Indonesian National Anthem Republic of Indonesia. The fourth is greetings by head of Stikes Alifah Padang. The fifth is photo session. The sixth is presentation from the speakers. The last agenda is closing. Welcome to the first Alifah Health Science College Symposium from Stikes Alifah Padang, West Sumatra, Indonesia. Hopefully, the activities run smoothly and knowledge can be useful for all of us. The Honorable Head of the Alifah Nur Ikhlas Padang, Mrs. Hajah Fatmi Arma SKM, and the Council members, the Honorable, the speakers who have joined us today, Mr. Tarun Kaswani, BSc, MSc, MPhil, PhD from Mass General Hospital, Harvard Medical School. Good evening, Mr. Tarun Kaswani. The Honorable Mr. Sohail Rana, MSS, MPH, PGD, co-founder Platform for Development Practitioners. Good evening, Mr. Sohail Rana. The Honorable Head of Stikes Alifah Padang, Ms. Dr. Nurse Asmawati MKep. The Honorable Stikes Alifah structural leaders, the lecturers, and all the academic staff. We would like to thank the committee who have done their best to carry out this webinar, and especially to all the webinar attendees, First Alifah Health Science College Symposium of Stikes Alifah Padang. First and foremost, let us express our praise and gratitude to God Almighty, who has bestowed His mercy and grace. Because of His blessing, we can attend the webinar this morning. And also praise to our Prophet Muhammad Sallallahu Alaihi Wasallam for his effort and to make a better life for humankind and knowledge. Ladies and gentlemen, in order for our webinar to run smoothly, let us listen to Asma'ul Husna.
Ya Allah, Ya Rahman, Ya Rahim, Ya Malik, Ya Quddus, Ya Salam, Ya Mu'min, Ya Muhaimin, Ya Aziz, Ya Jabbar, Ya Mutakabbir, Ya Khaliq, Ya Bari, Ya Musawwir, Ya Ghaffar, Ya Qahhar, Ya Wahhab, Ya Razzaq, Ya Fattah, Ya Alim, Ya Qabid, Ya Basit, Ya Khafid, Ya Rafi', Ya Mu'iz, Ya Mudhil, Ya Sami', Ya Basir, Ya Hakam, Ya Adil, Ya Latif, Ya Khabir, Ya Halim, Ya Azim, Ya Ghafur, Ya Shakur, Ya Ali, Ya Kabir, Ya Hafiz, Ya Muqim, Ya حسيب يا جليل يا كريم يا رقيب يا مجيب يا واسع يا حكيم يا ودود يا مجيد يا باعث يا شهيد يا حق يا وكيل يا قوي يا متين يا ولي يا حميد يا محصي يا مبدي يا معيد يا محيي يا مميت يا حي يا قيوم يا واجد يا ماجد يا واحد يا صمد يا قادر يا مقتدر يا مقدم يا مؤخر يا أول يا أخير يا ظهير يا باطن يا والي يا متعالي يا بر يا تواب يا منتقم يا عفو يا رؤوف يا أحد يا مالك الملك يا ذا الجلال والإكرام يا مقسم يا جامع يا غني يا مغني يا مانع يا ضار يا نافع يا نور يا هادي يا بديع يا باقي يا وارث يا رشيد يا صبور يا الله 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 يا الله.
Next agenda, let us listen to Indonesian National Anthem Republic of Indonesia. Ladies and gentlemen, next agenda. Welcoming speech from Head of Stikes Alifah Padang, Mrs. Dr. Nurse Asmawati MK. The time is yours. Bu As, suaranya masih di-unmute, Bu?
Okay. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, good evening, and greeting to all of us. The Honorable Chairperson of the Alifah Padang Education Foundation, Ibu Haja Fatmi Arma SKM, and the council members, the respected speakers, Mr. Soherana, MSS, MPS, PGD, from United States Agency for International Development, the respected speakers, Mr. Tarun Keswani, MPhil, PhD, Marsh General Hospital, Harvard Medical School. Welcome and thank you for accepting our invitation. The Honorable Alifah, Stikas Alifah Padang, structural leaders and lecturers and the inter-academic community of Stikas Alifah Padang, Committee Symposium, especially the symposium attendees. Alhamdulillahirabbil alamin. All praise is only to Allah Subhanahu wa Ta'ala, where we ask for forgiveness, protection, and help. Even though we are still in the second year of the COVID-19 post-pandemic, we are still given help and opportunities so that we can hold the symposium today. May Allah Subhanahu wa Ta'ala always guide and protect us. Salawat dan salam kita sampaikan kepada junjungan kita Nabi besar Muhammad Sallallahu Alaihi Wasallam. Ladies and gentlemen, with the leaders, committee, and academic community of Stikas Alifah Padang would like to express our gratitude and highest appreciation to all of the participants who have attended our symposium, because soon we will be listening to material in the first Alifah Health Science Symposium, Emerging and Remerging Infectious Disease, Future, Challenge, and Strategy. Hopefully, this event will be protected by Allah Subhanahu wa Ta'ala. Amin. Ladies and gentlemen, at the respective gates, starting in remark, all us to present to profile Stikas Alifah Padang, West Sumatra. Stikas Alifah Padang, under the auspices of the Alifah Padang Education Foundation, established in 2005, has a vision becoming a whole college with noble personality and national competitiveness in 2030. We are committed to continuously improve the quality of higher education beyond the national standard for higher education and continue to strive toward the international level. Currently, Alifah has exhibited an excellent accreditation rating from the National Accreditation Board Higher Education. In this moment, we are very proud to be able to create an academic atmosphere at Stikas Alifah Padang. The theme we will be discussing now is highly relevant to development of global health science. Thanks again to the speaker who are outstanding experts in their work, who are willing to share their knowledge with Stikas Alifah Padang. Thank you to the Alifah Padang Education Foundation for always providing moral and material support. Thank you, Committee Symposium and sponsor. Olahraga naik sepeda ke Khatib Sulaiman. Hari ini kita simposium. Selamat mengikuti simposium. Dengan mengucapkan Bismillahirrahmanirrahim, the first Alifah Health Science Symposium is officially opened. Billahi taufiq wa hidayah. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mrs. Dr. Nas Asmawati, for your opening speech. Next agenda is photo session. We ask all the attendees to activate the camera. Thank you. dapat kami informasikan kepada seluruh partisipan, kita akan berfoto bersama. Dimohon untuk dapat mengaktifkan kameranya. Terima kasih. Okay, we start from slide 1. 3, 2, 1, cheers. Okay, next slide. 3, 2, 1, cheers. Next slide. 3, 2, 1, cheers. Okay, thank you. Well, ladies and gentlemen, now we move to the main event, the presentation of the speakers. Number 1, Mr. Tarun Keswani, BSc, MSc, MPhil, PhD from Mass General Hospital, Harvard Medical School, as the first speaker. And the second speaker, Mr. Sohel Rana, MSS, MPH, PGD, co-founder Platform for Development Practitioners, as the second speaker. As we all know, our today's topic is about emerging and re-emerging infectious disease, future challenges and strategy. This session will be led by Ms. Febri Handini, MKM. To know, Ms. Febri Handini is Master of Public Health graduate from University of Indonesia. She is one of lecturers in STIKES Alifah Padang. She is active in several conferences as oral presenter. Okay, to Ms. Febri Handini, time is yours.
Yeah, thank you for the kind introduction, Ms. Fatmi. The respected speakers, the Honorable Bu Haji Fatmi Arma SKM as the head of Foundation Pendidikan Alifah Nur Ikhlas Padang, as well as the council members, the Honorable Mrs. Dr. Nurse Asmawati MKEP as the head of STIKES Alifah Padang, all the structural leaders, all the head units, distinguished guests, ladies and gentlemen, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Okay. I'm Febri Handini, and it gives me a great pleasure to be the moderator for the first Alifah Health Science College Symposium of STIKES Alifah Padang. As the symposium is part 3 of webinar series organized by STIKES Alifah Padang, and the theme for the symposium is Emerging and Re-emerging Infectious Diseases, Future Challenges and Strategy. Being the largest archipelago country in the world with a tropical climate and unique flora and fauna, Indonesia habitats one of the most diverse biomes in the world. These characteristics also facilitate the transmission of zoonosis and provide ideal living and breeding circumstances for arthropods, non-factors for viral diseases. And based on reports of the Ministry of Health, Republic of Indonesia, shows a significant increase in dengue infection incidents. And furthermore, chikungunya, Japanese encephalitis, and rabies are proven to be endemic in Indonesia. In addition to that, there is an increased risk of other emerging diseases, such as Zika virus, monkeypox, or Hanta pulmonary syndrome, since sporadic cases were reported around the region. And besides new diseases, tuberculosis, dengue virus, malaria, and diphtheria are continuously reported in Indonesia and classified as re-emerging illnesses. And in today's symposium, we will take a closer look at how the experts are responding and managing this issue. And before we begin, let me explain briefly the rule of the session. So we have a total of 45 minutes approximately for each speakers. And after all the speakers deliver their presentation, we'll follow by approximately 15 minutes for question and answer session. And all the attendees may ask questions by typing your question in the chat Zoom or raise your virtual hand and ask your question directly to the speaker you address, or as well as typing your question in the link that the committee is provided. And we will also remind you that selected questions will get door prize from the committee. Jadi Bapak Ibu dan peserta yang hadir pada hari ini bisa setelah nanti speakers, setelah nanti narasumber menyampaikan presentasinya, para Bapak Ibu dan peserta bisa langsung bertanya langsung atau bisa langsung bertanya lewat kolom chat atau bisa juga bertanya lewat link nanti akan disampaikan oleh panitia. This morning we have invited the speakers from United States of America and Bangladesh. The first speaker is Mr. Karun Keswani, PhD from Massachusetts General Hospital, Harvard Medical School. How are you, Mr. Tarun? Welcome to West Sumatra, Indonesia, virtually. And then,
thank you for the introduction.
Yeah, thank you. And the second speaker is Mr. Sohail Rana from United States Agency for International Development for Bangladesh. How are you, Mr. Sohail? Apa kabar?
Yeah,
Okay.
Thank you. Yeah, good morning. And I'm good, thank you.
Okay, thank you. So, ladies and gentlemen, first I'd like to read a brief introduction of the first speaker. Mr. Tarun Keswani, PhD, is originally from India, and he earned his doctorate at University of Calcutta, Kolkata, India, in the field of zoology. He is now a researcher fellow at the Center for Immunology and Inflammatory Diseases at the Massachusetts General Hospital, Harvard Medical School, Boston, USA. And he has held important positions in several institutions both in India and United States of America, as well as in France. He's also actively participated in several international conferences and engaged in significant collaborations. He is also a reviewer of several international journals and earned more than 10 honors and prizes, as the latest he got is postdoctoral fellowship from European Union Horizon. And he was also involved in studying virus-like mechanism in malaria parasite fitness, which leads him toward more than 20 publications related to malaria. So, ladies and gentlemen, without further ado, I would like to invite Mr. Tarun Keswani, PhD, as the first speaker. From Mr. Tarun, the time is yours.
Thank you very much for the nice introduction. I will share my screen. Is it visible?
Yes, sir.
And I hope you can see the full screen. Okay. Perfect. So thank you to the organizers, the moderators, and all the students. I assume it's Saturday morning there. So thank you for spending the beginning of your weekend listening to some of the boring stuffs. And today I'm going to introduce a well-known topic, emerging and re-emerging infectious diseases. As we all are from Asia, we know that in Asia, we are faced with a number of emerging and re-emerging diseases. And the most important part, which we learned in last couple of years, that the important aspect is how to prepare during some epidemics and pandemic situations. And I think we responded very well in the last couple of years. And the goal of this kind of webinars is to increase our preparedness, how we cope up with sudden outbreaks. And I think we did pretty well. So currently I'm working as a research scientist at Mass General Hospital, Harvard Medical School. Here I'm working primarily on the allergy-based immune responses. But previously I was working on malaria, toxoplasma, host immune response in mouse models and human samples from cohorts from several parts of the world. But my main goal in future, the area which I will pursue, is cerebral complications due to infections. As all of you know that now with the COVID is almost gone now, I will say, but there are several studies where they are saying that the brain is impacted with the COVID virus infections. So that will be my future goal, which I will do. Anyways, coming back to the topic, today's topic. So this geographical distribution shows all the emerging and re-emerging, most of the emerging and re-emerging infectious diseases, as the moderator was also speaking about the current infections in Indonesia. I would love to see the malaria rates now, what is there, because currently in India, dengue is more prevalent. So you can see that all the dots and the different symbols, they show, this is 2008 data. And as we are aware that the SARS-CoV was already there, but this time it was a different strain, and it's globally distributed. So the important aspect before we know about any new disease or how to control the emerging diseases in US and several parts of the world, there are several networks, how to distribute the information, how to get the updates about the new strains. So that network already existed before the COVID. And so that helped researchers worldwide to deal with this situation, I will say, in a short span of time, because normally for a vaccine to get developed, it takes 5-10 years. And even 80% of the vaccines don't pass the clinical trial, the final phase. So I will say that pretty well job done. So I distributed my talk into 4 sections: the emerging and re-emerging diseases, immune response triggered by vaccination, Ebola. I will discuss about Ebola because before COVID, we all are now well aware of all the COVID stuff, but before COVID, there was Ebola that was very deadly. And there were sudden outbreaks here and there. And in 2019, vaccine was developed by GSK and other companies. That time I was working on Ebola in France. And I can share some data in that. And then the emerging disease, malaria and co-infections, and basic and translation human immunology. So first, what is emerging infectious disease? So according to the National Institute of Allergy and Infectious Diseases, the emerging infectious disease are commonly defined as outbreaks of previously unknown disease or known disease rapidly, coming back in last 2 decades, like Ebola, SARS, or some infectious diseases which cannot be controlled, which stays. For example, as I mentioned, like dengue fever, right? It always persisted. It cannot be resisted by now. HIV infections. But then what is re-emerging infectious diseases? So re-emerging infectious diseases are primarily those infections which we thought at some point of time that we have controlled, like malaria, cholera, TB, but they evolve again. So those are re-emerging infectious diseases. Then what's the global response about this establishment of emerging and communicable diseases, and how to survey and keep track of the travelers who are traveling to disease-prone areas so that they stop spreading when they come back to the non-diseased countries. So this was long back in 1997. There were World Health Organization reports that even when I was in France, I saw that before I traveled to India for my vacations, we always had to get tested, that whether after coming back, whether we have some other infections or not. But this time, things went out of control, I think in 2020 beginning. So, and as the global travel has increased in last couple of decades, so there are more recent outbreaks and many of the newly discovered infections. We visit places which are totally unexplored, being unaware of the fact that what new potential hazards we can deal with, right, in different parts of the world, which we can't help. But what we can do, that we can discuss with travel medicine specialist or healthcare provider. I think this is a very good platform for all the students, They can guide their patients, and provide them the immunizations and preventive medicines before they travel. So if you summarize all this part, like how a new highly virulent parasite or virus coexist in the nature or evolve, because it's a triangle disease concept. Host, we know, is an important factor. The environment, where there are a lot of factors which change the ecology and biodiversity, which helps the pathogen to co-evolve. This shift in ecosystem exploit the new niches, which causes the new strain to evolve, and then the immune response. So all of you are well aware that our host immune response helped some patients to overcome some infectious diseases. For other patients, they were unable to cope up with the situation. So all depend on the microenvironment or the inbuilt immunity, which we gained through several years. So this complex and multiple interactions between immune mechanisms, pathogen, and environment as a whole plays an important role in this emergence or re-emergence of the new diseases. So this is a chart which was updated in 2020. You can see that how each emerging and re-emerging disease outbreak occurred, right? In 2002, you can see that there was SARS outbreak for 1 or 2 years, and then Zika virus, and then H1N1 influenza, then Zika again. If you see here in 2014-16, Ebola, there was a massive outbreak in 2014-16 in Congo, and then the outbreak again happened here, and MERS always persisted in Southeast Asian countries. And then COVID-19 started from 2020, and we still know that it still persists. So this is a summary how disease goes and comes, and so taking important measures beforehand so that the spread is being restricted to some extent. And this is also related with the global travel, which I was discussing with. So now we will discuss the immune response part triggered by Ebola vaccination. And as I was discussing, that there was a sudden outbreak last year in 2021, but that was not in limelight because people were busy with the COVID. And due to the vaccination in 2019 and all, the things were controlled to some extent in 2021. So how this virus spreads and what are the sources? We know that it's mainly spread from Africa, and the fruit bat and the gorilla, chimpanzee, all the wild animals are the possible source of that, and how it transmits. So the possible source is by close contact with the different parts of this infected animals, consumption of those meats, and touching objects which were in contact with the virus. And it causes a severe damage from 2 to 21 days. It damages the hepatocytes and the other immune organs, and it also damages the blood vessels. So this is another representation of that. So it's very deadly viral disease, and most of the patients die. And if they recover that first couple of weeks, and then it's possible that they can cope up with that. But now there is possible vaccine, so it's good to go. So I will not go into detail about this vaccine studies and how they are designed before going into clinical trial and then being tried on humans. But there are a couple of glycoprotein and nucleoprotein, which was designed for this study, what I was doing in France, in Nice. And so for each of this, like the Ebola glycoprotein, it has a long B cell response capacity, and the Ebola glycoprotein and the nucleoprotein in combination, they had a strong T cell response. For a good vaccine, the delivery and the boosting is very important for the good vaccine, immune response. So this is a primary schema to show how the priming and boosting helps. So the formulation is very important, the antigen dose is important, and the route where the vaccine is administered. the formulation is important because the retention of the immunogen at the site of injection is very, very important. And so that it helps the innate immune system to activate the maturation of dendritic cells as well as other innate nucleocytes. And finally, if that activation happens in a proper manner, then the antigen-specific immune B cell T cells gets activated. So if you see this diagram, like a vaccine, when it enters, it gets primed by dendritic cells. And this is a whole picture. So it gets primed by dendritic cells. And then it gets engulfed by the CD4 T cells, which secrete cytokines, and CD4 T cells also differentiates into different subsets like Th1, Th2, Th17, and Treg cells. These are different subsets of T cells. And also that leads to the Th follicular cells, which further helps in the antibody generation, the long-lived plasma cells. Then the antibodies are directed in the blood and it migrates towards the other regions like the bone marrow, and then that helps in the generation of the memory response. So memory response generation is very important for any vaccines, good vaccines, because when the person will be infected again, that memory response will boost the immune response and will help the person to recover better. So how this works, so it's an example in mouse model, because before we do human trial, people do research in mouse models. So I did that when I was in France, and we selected the antigens and the peptides and the adjuvants, which helps to retain the antigen at the site of injection, and also the emulsion, which helps in slow release of the antigen. So we inject the mice at the 0 day of infection, and then so these are two different studies. So here the glycoprotein and the long peptide was added. Then after 14th day, there was second dose. So I think you can correlate what is going on with the first and second dose these days, and then the booster. So after the second dose, 14 days later, you can see the effector phase, where we studied the serum cytokine level, the B cell and T cell response, and the cytokine staining cells, and the IFN gamma response. Because the IFN gamma response, better IFN gamma response, it shows a better effector phase. And then later on, we again studied on the same mouse, separate group of mouse, but they were started in the same time. And there also we studied the memory B and T cell marker and the IFN gamma studies. So what we found that a strong B memory response was observed at the end of the study, and strong T cell memory was observed. So to make long story short, for a good vaccine to work, a strong B and T cell memory response is very important for a long-term memory response, right? So as I was discussing, so cytokines releasing at the initial phase, expansion of CD4 T cells, and then the differentiation into different T subsets depending on the vaccine formulation or the vaccine prepared for a specific disease. A specific T cell will be differentiated, and then the long-lived plasma cell, which helps in the preparation secretion of the antibodies, and which helps in the memory T cells and the central memory T cells, which makes a vaccine much more stronger. And then it goes to the phase 2 and phase 3, where it gets tested into small cohorts of patients. And if the study data is good, then it goes to the market and people get those vaccines. So for a protective efficacy, a strong memory stimulation and long-lived memory antibody response is very vital. So if a research doesn't verify that which B cell population is stimulated, whether that B cell population is long-lived in our body, then the efficacy will be very less. So it's a general diagram to show how to target, the memory B cell versus non-targeted vaccination. Because you might have seen that there are a lot of vaccines from different companies, but some are only used like the Pfizer, Moderna, and there are a couple of other. So because they have long-lived memory response, though scientists are still working to get a more longer memory response. So I think in a couple of years, we will have much better results. In summary, for any memory T cell subsets, I discussed the B cell now. For the T cell subsets, if you have higher IL-2, a medium IFN gamma, and the less cytotoxic effect, that is better, like then central memory and the T stem cell memory. This is called T stem cell memory. Then a vaccine is suggested as a good vaccine. If they have low IL-2, high IFN gamma at effector phase, and cytotoxicity is high, then it's not good. So now I will discuss. So if you guys have any question, if any professor or any moderator have any question, please feel free to interrupt me anytime. I will be happy to take questions. So now I will discuss the emerging disease like malaria and other co-infections. So I will discuss toxoplasma along with that. So before moving to malaria, I mean, it's not that I was working on malaria. I'm still connected with a lot of malaria journals and all, but it was very shocking to me last couple of years that because all the healthcare staff, the facilities, the administration, they were so busy, which is quite natural with the COVID stuff. A lot of World Health Organization projects or a lot of local administrative things which helps in controlling the malaria or other parasitic infections were somehow hampered, right? We all are aware of that. And as we know that the malaria is mostly impacted to kids, and the potential impact of this health burden is very worrisome. In last year, in 2020, there were cases where patients were treated for COVID-19, but they had malaria because they traveled to some places and it was not well taken care of because as I said, everything was focused on COVID. And this is one case study where the patient died with the COVID. Malaria. Initially, she had malaria, and then in the emergency room, she got COVID-19 also. So, as the immune system was compromised with malaria already, so the patient was unable to recover with the COVID-19. Even they got admitted to the emergency department. So, this is an example of co-infection, right? So, estimated cases till 2018 was 228 million. Among that, I mean, in one child dies every 2 minutes from malaria in Africa, which is very, very shocking, right? And most of the symptoms which are deadly symptoms are the cerebral malaria, the respiratory distress, and other stuff, though preventive measures are there for malaria, but it's tough to still control. There is no proper vaccine yet. So how the immunology of malaria works, like, because the Plasmodium parasite and host immune system, they plays a very tenuous balance. I mean, it's tough because the parasite, the malaria parasite, I will explain later on, it stays in mosquito also and also in the host, and there is also dormant phase. So it's tough for the vaccine researchers to design a vaccine for a specific stage of the parasite. And as also we get the symptoms when the parasite is already in the blood, because when it's stuck in the liver, we don't get any symptoms. So also it's difficult to understand the mechanisms of protection or pathology in humans. So that's why we need mouse model, which I will explain later on. So if you try to understand the mechanism or the life cycle of malaria parasites, mosquito for sure plays an important role, and it injects the sporozoite via bite, and it enters the liver, and then from the liver it ruptures and the metazoite released, and it comes to the blood stage. And in blood stage it replicates again, and then when the mosquito bites human infected with malaria, then it gets the ring stage or the gametocytes and other stuff, and it again replicates. So this is a cycle. So to prepare a vaccine for malaria, it's very complicated because you don't know at which stage if you prepare vaccine for this stage, then you don't know if the parasite is already there in the liver in the dormant stage. So that's why it's very complicated, and also it's complicated to control the mosquito, because that will also sometimes disturb the ecosystem, right? So scientists are working on all the aspects, and I hope we will soon get something good out of it. So as I mentioned, that it's a re-emerging disease, and it's a vector-borne infectious disease caused by protozoan parasite. And according to disease severity, the different strains of malaria are ordered here, like falciparum is the most severe, then also vivax, and then the malaria. So according to WHO definition, cerebral malaria is there should be a coma, and the peripheral parasitemia present of peripheral parasitemia. So as we discussed about cerebral malaria and the immune system, then how the central nervous system works in the parasitic infection. So this is a close view of this part where the lymphocytes, the monocytes, the neutrophil, they are outside the blood-brain barrier, and inside the brain, you have astrocytes and microglia in a normal state. But during the acute infection, Plasmodium falciparum, there is a breakdown of this blood-brain barrier, infiltration of the T cells into the brain, acute inflammation, and all the cytokines play an important role in the disease progression. So in detail, this is the endothelial cells in the brain, and this blood-brain barrier breakdown caused due to the clumping or agglutination or rosseting. These are all clinical terms. And these are the parasites. This is normal RBC. So the parasite accumulates in the blood-brain barrier, and it leads to the obstruction of blood flow. So the oxygen flow is less. It leads to the hypoxia and release of toxins, and all the leukocytes and platelets leads to the release of the cytokines, and it causes multiple seizures, neuronal damage, which ultimately leads to coma. All because this leads to the blood-brain barrier breakdown. So what are the key questions? So what is the pathophysiology of severe disease? Also, there is one interesting question, that how do people living in malaria endemic regions become protected from severe disease? Because it's not that everybody gets infected with malaria, or even after infection, everybody don't develop the disease. So there is some protective mechanism in certain humans, right? In the malaria endemic regions. So what's that? So all these questions are tough to answer with the observation by infected humans. So we need some mouse models, rodent models, because patients who died with cerebral malaria, we only get the terminal data, end time point. And also there are a lot of ethical issues, the review boards, human review board to get cleared with some of the research work. So this animal model helps, because there you can also infect the mouse with the mosquito. You can see this picture where you have mosquitoes here, and they infect the mouse, so that you reciprocate what is happening in nature, and then do the bench work to understand what immune mechanism is going on. So this is one example where if you knock out this is Burgee resistant to gene, you can see that after infection, this is a wild type mice where there was death around 7 to 8 days post infection. But in a resistant gene knockout mice, the mice survive like 30 days and they don't die with the cerebral malaria. So I will explain the cerebral malaria in further detail. And the parasitemia was one of the factors which we have to measure because this is one of the important parameters. And you can see that here the parasitemia is increasing as the mice survived, but they don't die with the cerebral malaria, they die with hyperparasitemia. You can see the difference during this phase in parasitemia. So, splenomegaly is an important symptom during malaria. So you can see that this is a wild type mice. It's this control spleen. This is infected spleen at 6 to 7 days post-infection. And this is the resistant mice control spleen. And this is the infected spleen that gets bigger, because splenomegaly is one of the parameter where the spleen is trapping all the infected RBCs and trying to help this host to overcome that. So what is happening in brain? So these are mouse brains. These are controlled brains. They are normal. There is no blood-brain barrier disruption because there is no infection. But if you see this brain, why this is blue? Because we injected a dye, a retroorbital injection. And if there is a rupture in the blood-brain barrier due to parasite infiltration, then there will be leakiness in the brain, right? And the dye reaches the brain. So we sacrifice the mice after injecting the dye, and we can see that the brain turned blue. But this is a resistant mice after infection, right? And the brain is still similar to control. That means the blood-brain barrier is intact. So, I mean, the summary of this study was that this is a helpful model or gene to knock out and to understand the mechanism. So what is going on? After infection, so this is a schema during the 6 to 7 days post-infection, because that after that, the wild-type mice is dying. So this, I'm not showing all the flow cytometry data or immunological data. I'm just showing the schema to help you understand what is going on in a wild-type mice where the mice is dying due to experimental cerebral malaria. So the chemokines and the T cells all are migrating to the brain, and the parasite accumulation is huge here. You can see that the monocytes and other stuff is all going towards the brain during the peak phase of infection, and which leads to the blood-brain barrier disruption. And that is also causing the brain resident cells, the astrocytes and microglia and the monocytes, to hyperactivate and leads to inflammation and edema, swelling in the brain, and finally leads to death. And what's the scenario in the resistant mice? In the resistant mice, the spleen gets bigger, as I showed you. The T cells and the chemokines and the cytokines, they are all retained in the spleen. The migration is less compared to the wild-type mice. Parasite sequestration is less, as we also confirm with the brain image, you can see, but also we did the immunophenotyping of all these immune cells after taking out the spleen and processing it for flow cytometry. You can see that the microglia, astrocytes, and monocyte activation was less. This shows that they were all inhibited or less expressed. And this mice didn't die with the cerebral malaria, but later on they died with the hypoparasitemia. So, yeah. So now I will move to the co-infection part. So toxoplasmosis, as we are all aware of, they're very much in nature, and it comes from the rats, the cats, and all, and from the other meat products also. And in USA and in France or in Europe, it's very much persistent. I don't know how much we are aware of the toxoplasmosis in India. I have no idea, and maybe I will get some feedback from professors there. What's the situation in Indonesia about toxoplasma? So in toxoplasma also, during the acute infection, there is sequestration in the central nervous system, disruption of the blood-brain barrier, but I will show you it's different kind of blood-brain barrier disruption, acute inflammation, and also this activation of different cells play an important role. So I'm not going into the detail of toxoplasma, but what I'm going to say that during toxoplasma infection in mouse model, if the mouse is already treated with toxoplasma and then we infect with the Plasmodium infection, which I showed previously, then the mice don't die also. They survive that phase, because if you see here the green line, the purple line, and the red line, these two are the controls, right? So you have treated parasite. So the parasite is treated with sulfadiazine, and then the treated group. In all these three, because there is a Plasmodium infection, they are all dying. But in this yellow line, it was infected beforehand with toxoplasma, which also goes to the brain. That presence of toxoplasma in the brain is somehow helping the mice to survive the infection with malaria. So this co-infection is helping the mice to cope up the cerebral malaria. So imagine, a few slides back, I was saying in endemic region, there are other infections also. I mean, co-infections. So my question is that, does co-infection helps people from, endemic regions to cope up with deadly diseases? So the answer is possibly yes, because there are several studies in Southeast Asian countries, also in Singapore and also in African countries, where they have found that coexistence of viral or other parasite infections helps people to cope up with one of the deadliest infections. So this is another image where this two you can correlate that previously I showed that this is a control brain and this is the malaria infected brain in mouse model, and this is a toxo. So you can see that there are some small segregation here because due to toxoplasma infection, there are cysts in the brain. But if you see this co-infected, means it was infected with toxo and then it was infected with malaria parasite. So the brain do turn a little bit blue due to the cyst and the parasite, but it's significantly less disrupted than the malaria strain. So here, other immune cells play an important role. But my previous lab, now they're working on this. They found out that the autophagy and the T cell exhaustion and the antigen presentation might play an important role. What is happening in brain? I'm not giving further details in this work because that is we are still working on this to publish. So the last chapter, basic and translational human immunology. So how we translate the work from lab to field, or from field to lab, or connecting global labs or global research centers to make things happen. That's why that was a good example. The COVID was a good example that how different labs worldwide coordinated and made that happen. So now things are changing, and there are advanced tools for basic and translational human immunology, like blood-based immunoassays, the reporter assays, modeling immune tissues. Like modeling immune tissues means organ on chip. We are all aware of the term chip. Now people are working on organs modeled on chips. There are organoid cell cultures and in vivo studies like the mouse models, in silico models and bioinformatics, natural immune variations. All these factors are important to understand the wealth of the human data for translational immunology. Like cohorts are very important, as I was mentioning before, that it's tough to understand all the aspects just only studying human sample. But there are several groups also in Europe and in USA where they're trying to reduce the use of mouse model or other models due to animal ethics issues. So now people are trying to understand how to get more information from cohorts, from the specimens, from the tissue samples, from the fecal samples, and to understand the specificity of the genetic recombinations, understanding the phenotype via immune cells from the PBMCs, also get the functional aspects like in vitro assays to understand the cytokine response or the immune cell functions or the memory functions, and also the environmental factors from the gut microbiota. Combining all this information is the future of translational human immunology, because previously what we were doing, we were working on mouse models and non-human primates and testing in humans, right? Currently, what we are doing is we are gathering information from the human data or samples from the patients who are getting infected with the different infections, whether it's re-emerging or emerging, and then trying to study further in animal model and in vitro data to gather more information, and was correlating and predicting with the help of bioinformatics. But the future, according to this, it's nice review published, I think 1 or 2 years back, from the mass cohort collection, because now many people have this sample from the COVID patients that will help the computational biologists and the in vitro models and the ex vivo stuff to get a brief idea and reduce the use of other model systems for the translational human immunology approach. So in summary, the re-emergence may happen because, of a breakdown in public health system, like it may happen for malaria, I don't know, that were once under control. Like in Calcutta, India, I got the news that the dengue prevalence is prominent now. On the other hand, co-infections may promote intricate and complex mutualistic interactions with the host immune system. I gave you one example with the toxoplasma and malaria. But there is no quick and simple experiment, that can firmly determine whether immunity will be effective or lasting, So it's just too soon to know what I feel. So these are some pictures of my previous labs, and this was in Europe. This was in New York, and currently I'm here at Harvard Medical School, MGH. I would love to thank you all for your patience, and I hope I didn't bore you a lot. If you have any questions, please feel free to ask. I will try my best to answer. Thank you.
Well, thank you very much, Mr. Tarun. It's a clear and interesting explanation, and there are a lot of lessons learned from what you explained, such as eventually traveling mostly can affect the incidence of emerging diseases. Yeah. And many of the newly discovered infections have actually been in existence for a long time. And many travelers also unaware of the potential hazards in different parts of the world. So I think that immunization and take the preventive medicines is obligation now before you have to traveling around the world.
Yeah.
Okay. Thank you very much for Mr. Tarun. And I think that we have to continue to the second speaker before we move to the next session. And now we will continue to the second speaker, Mr. Sohail Rana, MPH, PGD, PMP. Mr. Sohail Rana. Okay. So Sohail Rana comes from Bangladesh, and he earned Master of Public Health from Gajah Mada University in Indonesia, and completed his graduate diploma in project management professional from Bangladesh Institute of Management Studies under Qualify UK. And Mr. Sohail is now a project management professional with over 7 years of experience and specializing in the areas of health system strengthening, primary healthcare and service delivery, equitable access to essential health services for all, research, and many more. And he has virtuous hands-on experience on leading the capacity development activities for different stakeholders, project implementation, capacity development, sectoral approach for emergency response. And Sohail is currently working for USAID's Local Health System Sustainability Project as Capacity Development Coordinator to enhance capacity of local government. And he also worked for the World Health Organization for emergency response for Rohingya refugees, and also as a field health sector coordinator, Bangladesh Nutrition Activity Project and Johns Hopkins University for different public health project and implementation. Mr. Sohail also creates more than publication related to universal health coverage. So without further ado, please welcome Mr. Sohail Rana. The time is yours.
Yes, yeah. Thank you, moderator, and thank you, the participant and the organizer, for inviting me for the session. And thank you especially for the Rajian who actually reached out to me and just offered me for this one. And let me share my screen. So please confirm my screen is showing.
Yes, Mr. Sahel, it
Oh.
works.
Okay. Okay. Thank you, and thank you, my previous the presenter, the Tarun. As yeah, it was very nice presentation, and I will for my presentation actually I will try to more focus on the health systems for this infection disease controls. So there's some the overlapping with the previous one, but I will try to avoid that one. And apology for the if there is any overlapping, I am apologizing for that. And please stop me anytime in my presentation. If you have any question or query or any suggestions, please stop me. I'll be happy to respond to your question or query or participate in the discussion actually. And for the participants, yeah, you are also welcome to ask question in the Bahasa Indonesia. I can speak Bahasa Indonesia as well. So please let me know if you are not comfortable in the English. Thank you. Okay, so for today's topic it's we already know. So it's about the infectious disease, the emerging and emerging infectious disease, and I will more focus on the health system approaches, how we can control it. This is the a bit content about it. We will try to focus on these items, but briefly. So in the background, so I can say that in the globally, people at risk of the life threatening due to this infectious disease and this acute and the chronic infection actually. And it was actually now the condition is much wider than before, and due to the social, political, and ecological changes in the 21st century. So compared to the other centuries, the prior centuries, now the problem is more spreaded and more articulated. And the globally, it's about the 15% of the deaths actually is occurred due to the infectious disease in globally. So that it means that the globally the infectious disease contributed a good number of the cases for the mortality. So this which is also alarming for us. And for this controlling or the prevention or the other things, so actually the system biology is a holistic characterization for modeling of a living systems of biological network actually, which actually the previous presenter tried to focus actually this on the system biology. So it is very important for controlling this one. And this approach, the some reduction approach will be helpful to simplify or isolate the impact of the single components for the biological process, and which is also able to provide a comprehensive model for processing a quantification of all observable components of their relationship. That means there is the linkage of the relationship which will be helped by the reductionist approach for this one actually. And this is actually the I will say that some background actually. This is the epidemic this epidemic period associated with the different era. So this is the different eras, the epidemic period. So just like this post Columbus contact, then the classical antiquity, the trade wire, then the red one is the more the transatlantic slave trade, and the last one is the international air travel. So this is all are the medium, and by this way the epidemics were spread all over the world in the different eras. So this is the example of the epidemics period associated with the different eras of human transportation, the land, maritime, and the air travel. And also the overland trade networks and wire campaigning are thought to have contributed multiple epidemics in the Mediterranean in the late classical antiquity, which is shown in the green actually. And the beginning of the Antony plague, which reported claimed the life of the Roman emperor, the Lucius Varus, yes, Lucius Varus. The Mediterranean transportation red and gray actually leading the European contact with the Americas and the subsequent Atlantic slave trade resulted in the importation of the Plasmodium falciparum, malaria, and the novel pathogens. And the modern time, the air travel, the purple one actually the showcasing the importation of the severe acute respiratory syndrome, the SARS, MERS, coronavirus, the anti-coronavirus. So this is actually these three historical actual eras for the epidemics actually. And this one actually the infectious disease current status. That's a bit the statistics about this. So in recent years we saw the increasement. See the increasement increase of the air travel, trade and urbanization global. And the regional level, the also the accelerated the indicates the even more frequent transportation of the people and the goods between the growing and the urban areas. So that's you can see the global one and the regional one, about the infectious disease current status. So this is actually the alarming, and this is all are in the increasing trend actually, and this is the including the emerging and the emerging infectious disease. So if you see this, the air travel also increased, urban populations also increased, so which are some risk, increase the risk. The exports goods also the increasing due to the demand actually. So this is actually enhancing the emerging and the emerging diseases all over the world. And this is actually the death actually, the log deaths from the major epidemics from the 21st century. So this is from the 21st, the SARS, dengue, cholera. Then the 2010 is about the swine flu, measles, the MERS, the Ebola virus, the Zika, then the Ebola virus again, and the COVID now still is going on. So this is the trend of the emerging and the emerging diseases in the current time. And this is the disability adjusted life years. So it says the disability adjusted life years lost of the infectious disease. So that's like the SMARS, Middle East SARS, respiratory syndrome SARS, the neglected tropical disease, so etc. Actually in the disability adjusted life years, and it's all about the in billions. So this is the current situation of the infection, the reinfection, emerging infection diseases. And as we find out, this is one of the major health challenges or a threat for us, for the global people, and the not only the people, is also it's called now the One Health approach. So this is not only the people, it's also related to the animal actually. So it's not about the care about the population, but in the same time we also need to care about our animals actually and our environment to make sure the healthy lifestyle actually. And also this is the comprehensive approach for the One Health actually. And so this application of system approach is to infectious disease to a particular complex, which will involve the consideration two main particular components, the host and the pathogens. The which also told what stage actually. The host and pathogen is the main components of this one. And the well-designed system experiment can work effectively to model the model the ins and outs of the complex systems. So which will be able to capturing the the established knowns as well as the revealing novel unknown mechanisms. So that's like this. Some are re-emerging. So which is already known, which is the reason of the disease, but some are the not unknown actually. So this is the unknown mechanism of the emergent of the properties. So and this is the interdependence of the host and pathogen. So how they are the interdependence, the host and pathogen are interdependent actually. This is the relationship between the host and pathogens that dictates the outcome of the infections on the spectrum between the health and disease. So this is the very important to know. And the pathogens actually affect the directly changes on the host. So pathogen has a direct impact on the host, and that will be the elicits the response of the infections, which will be influenced by the underlying the environment. So that is about the environment as well. And the influence dictates the susceptibility, the virus resistance, tolerance versus pathogenicity, immune response versus immune deficiency, the virulence versus the non-virulence, and the ultimately health virus disease actually. So this is the all are the interrelated. And the system biology approaches are especially valuable in the infectious disease research, which is a way to capture the comprehensive picture of this interact the relationship. So, and in the picture you see the environment, which is also impact on the human being, also the pathogens, actually the host and the basically host and the pathogens, and the host and pathogens are also the interrelating, the replicating the rewiring the relationship. So this is the health and the disease. The red one is the disease. So this is all are the interrelated and all about the environment actually. And this is the system biology framework. So these three major things: the discovery, the representation, the application, the visual representation of the steps. These three steps outlined to review as part of the system biology approach to infectious disease, from the discovery to the representation and the applications to applications. And the arrows for the each component actually the focusing on the iterative and the interconnected nature of the system biology as a process. So for discovery, so there is to discover something new. So the data collection, data analysis, validation, and the experimental design is very important. Representation, the data, the representation of the network model, network analysis is very interrelated. And the applications is the we need to translate the data. The hypothesis need to develop a hypothesis, and the data dissemination, the reporting. So these all are interrelated. And this is not a single disciplinary. So this is the interdisciplinary. We need the interdisciplinary collaboration because this is not a single discipline to control about this one. So for system biology is the inherent team science, requiring input and the expertise from the diverse background to build the reliable and the informative model. So it's all about the comprehensive one. And it requires the collaboration across the disciplines. So we need the strong collaboration among the disciplines. And finding the domain expert. So we need to find out the domain expert who can share the excitement for the project, bringing together the team, sharing the knowledge, experience, and thus gathering the start and the incredibly challenges. And the principles of the interdisciplinary collaboration is in 3 steps actually: the partnership, organization, and the communication. The partnership will build the trust, respect, and the most and the share their ownership and the responsibility for success of the project. And the organization will ensure the how organized collaboration can play a vital role or crucial role to determining the ultimate success of the project. And the communication is very important. Crucial open and the regular communications is critical and success collaboration. So these all are the three principles for this interdisciplinary collaboration. And this is something is about the human social and the behavior factors actually, which is related to this emerging and the emerging infection disease, and they have the impact on it. So this is the biological factors and the in the environmental factors, and in the human social behavior factors. So is there three things: the pathogen, host, and the vector. So this is the very important: the pathogen, host, and the vectors, and this is the mutation and the transmission. So this is important, and every aspect have the detailed one actually. And now this is the before one system approach. Now the system thinking for combating for the infectious disease. So this is all about the system thinking approach. Is mostly two fundamental concepts: the complexity and the interiority. The complexity and the system complexity is generated from the structure of the integrated components, which will be able to explain how the constituting components are organized and interact with each other, and as well as with the environment. And the system thinking will offer a novel comprehensive perspectives, which will be examine the process of infectious disease transmission as a system with its structural complexity as a behavioral entirety. So this is focusing on the most entirety. And there is a complex approach for these principles actually. So the complex system approach is a holistic approach. So as we all know. And which will be intended to be a model, characterize, explain, and predict a behavior system actually. And it will be constituted a complexity of a complexity which will be drive or compute by using a conventional top-down reduction approach. And this system model, this complex system approach, three models basically: the system modeling, system exploration, and the problem solving. So system modeling will focus on this is actually the blueprint of the framework of the abstracted of the replicate from the real world observation. This is the system modeling based on the real life experience. And system exploration, which will be able to set the analytical tools that are devoted to understanding your operating mechanism underlying a complex system, and furthermore finding a explanation and making prediction about some observed system dynamics. So this is the system approach. And problem solving basically emphasizes the ability of the complex system approach to find out its own way to accept the adaptive solutions. So the system problems will be there. They will find out something which could be show the way about the solution, and which could be adapted from the other system as well. So this is the complex system approach in practice. Before one is this one is in the principle, and this one is in practice. So in practice there is three things: the analytical interface, the conceptual modeling, the real world grounding. So this all makes the practice actually. So, and this is the interrelated. So the problem driven conceptual modeling is this will actually translate the first time the real world problems in view of the epidemiological domain into a conceptual model, into a or a theoretical or the computational domain. And the data oriented real world will grounding the concentrates of the on discovering where the embodiment of the conceptual models through the model of the parameterization. And the last one is the goal directed analytical interface, which will devote it to further developing analytical method and the solutions. And these four essential steps: the conceptual modeling, real-world grounding, practice, and the analytical interface for the performing the complex system in approach in combating the infectious disease. So these all are in the practice. So this all are in the practice, and apart from this all practice of the, there is a few more approaches actually. So few more approaches. The One Health approach, which I told. So One Health approach, the point of entry approach, the clustering approach, the early warning system approach. So this is also the another things, another systems to control this emergency. The One Health approach actually is very close contact. It discover the close contact between the peoples, animals, planet, and their surrounded environment actually. And this will also emphasize the host characteristics, environment condition, and the habitat of the disruption. And this will be they will be more responsible for the existing the new diseases like bacteria, virus, fungus, protozoa, just like this. And this infectious disease is the essential constraint for the increase in the human, animal, and the environment interaction. So this is the essential constraint. And we need to identify the hotspot for the intervention of the or the prevention actually. So when we will take the prevention measurement, just like this COVID-19. So everywhere, every time we find the hotspot actually. So from the China, and it's spread to all over the world. Even I work for WHO in the Rohingya community. They're one of the biggest refugee slum in the world. So we also find out the hotspot about this infectious disease. So it's about the COVID or the diphtheria or the cholera or even the diarrhea. So first of all, we need to identify the hotspot and make the intervention based on that. So that that cannot be spread outside that hotspot. That we call the clustering one actually. So need to cluster identify the cluster one, the hotspot, then make the intervention actually. And after successful infection disease control measurement must be based on the understanding of the transmission of the pathway. And the application of the One Health method is the responsive approach for the infectious disease control. And this is actually the One Health approach for this controlling the and the what I call the early warning, alert, and the response system, the UART. So basically this is used in the mostly in the emergency response actually. So to increase the risk of the transmission of the infections, disease, and the other health conditions, the severe malnutrition, and the other things actually. So we established this UART systems in the humanitarian settings to get the daily notification about the disease trend and the treatment trend from this the locality actually. And it helps actually to see the disease trend and the cases, number of cases, and make them and define them as the outbreak or not. And this is the surveillance. This is the strong surveillance system is essential to detecting the disease outbreak quickly before the spread, cost the lives, and become difficult to control. And basically, this work is developed with the WHO is designated to improve the disease outbreaks detection in emergency settings. And UART is actually deployed during the emergency as an adjunct to the national disease surveillance system. So we are not telling that we don't need the national disease surveillance. Apart from the disease surveillance system, the UART is used to the commission for the further level of the checking of this outbreak actually. And basically, in that terms, WHO used to work with the Ministry of Health of the respective countries and to establish this system incorporate with the national system. And this one of these point of entry. So you all may know about the point of entry. Basically, the point of entry is something for the international travel. So in the earlier of this presentation, I saw the international travel. How this international travel are contributing the spreading of the infection, the infectious disease actually. So actually, the International Health Regulation 2005 basically they focus on the point of entry. So just like this, the COVID-19 was spread in the China. So if it was possible to control the point of entry actually, the exit and entry, then this COVID-19 may not spread all over the world basically. So it was spread all over the world through the travelers. So actually through the travelers. So for that reason, if it was able to make the point of entry screening more properly and take the initiative more efficiently and timely, so that may possible to prevent that one. And this promoted demand. This point of entry actually promotes the demand of the detection and management of suspected case at the entry or exit points of the country, including the port, the land port, the seaport, the airport, ground crossing, everything actually. And this management of the illness of travelers at the international port, airport, ground crossing, in the context of the COVID-19 outbreak, actually is objective to detection of the ill travelers and ensure the proper intervention for the ill travelers. They interview the ill travelers to determine the possibility of the symptoms and the exposure of the virus responsible for the respective disease, and the reporting cases for the suspected COVID-19 infections. And based on the trials and the other things, actually need to isolation, initial case management, and referral for those with the suspected COVID infections. So it could be the different referral mechanism should be in place based on the patient condition. And the clustering of the disease. So I already told about the clustering of this. When the one disease was found, actually we need to find out the cluster of this of the disease and make the disease within the cluster. And by then the disease may not able to spread outside the cluster. And when it is the clustering the disease within a certain area, need to proper intervention about based on the disease find out actually. And you need to ensure intensive response cluster areas, just like the disinfectant the places, distribute the medications based on the disease actually. For the diarrhea actually, so cholera actually. So what we did in the Rohingya response, so we make the we mapping the cluster actually, and we do the disinfections in the whole area, all potential sources of this cholera actually. And we also provide the medication that is the water purifications and the other things, the hand washing, the interpersonal communications, the IPC, infection prevention control measurement with them. So this is actually the clustering of disease and the things. And I think I'm end of my presentation. So yeah, this is the end of the presentation. And thank you. And please have any questions. If you have any questions to me, please let me know. And over to moderator.
Well, thank you very much, Mr. Sohail, for a unique and remarkable representation. And if I could add some highlighted point, a representation of the elaborate relationship between host and pathogen that ultimately dictates the outcome of infection on the spectrum between health and disease. And I think that we really need to do of interdisciplinary collaboration, such as partnership. The collaboration should be built on trust, respect, and most importantly, share ownership and responsibility to the success of the reducing emerging and re-emerging disease, and as well as organization and communication, to open and regular communication is critical to successful in handling infectious disease. And I think that we need to do One Health approach as one way to decrease infectious diseases, and close contact between people, animal, plants, and their shared environment provides more disease transmission opportunity. And I think that host characteristics, environmental conditions, and habitat destruction may lead to the spread of existing and new diseases. Ladies and gentlemen, okay, we have reached at the end of our main session. After this, we will continue to question and answer session. We already have several questions from the question link. We are glad to receive so many questions, but we are going to select the 3 questions first. I think the committee should share the question. We wait for the committee to share the screen first. Wait for a moment. Okay, this is from Ibu Desi Sarli. She is the vice head of Stikes Alifah Padang. Mrs. Desi Sarli asked, I think this is to Mr. Tarun, the entry of the first monkeypox virus in Indonesia brought by Indonesian citizens who had just returned from abroad. And this is threatening the transmission of the monkeypox virus in Indonesia. And how long is the incubation period of the virus until symptoms appear? And what are the strategies to prevent transmission of the virus? To Mr. Tarun, please.
Yeah, thank you, Ms. Desi, for this pertinent question, because in Boston, Massachusetts also we had monkeypox virus. So the transmission is not same as for COVID, the transmission rate. It only transmits with a close contact, or if you are touching the places, because it's not transmitted through air also. So it can be contained very well compared to the COVID, because it don't transmit through air. And the incubation period, if I'm not wrong, it's I think couple of days until the symptom appears. And the strategies is preventive medicine and also close door treatment, like, to separate from all the people. And I think all the hospitals have these facilities even before COVID to contain the monkeypox virus. So it's I will say it's not threatening, but you cannot stop the transmission of virus, because if people have to go to work, if people have to live a normal life, then people have to travel also. So you cannot, you have to coexist with viruses. So, but the monkeypox is not as transmissible as COVID is. Thank you for the question.
Okay, thank you, Mr. Tarun. Okay, I think this is the next question from Ms. Lala. I think this is to Mr. Tarun Kaswani again. Malaria and dengue fever are one of the recurrent infections diseases and generally occur in Indonesia, and the changes in the temperature is one cause of this disease. So does the manufacturer of malaria vaccine also consider the climate change for the occurring of the malaria?
Yeah, that's a nice question. I will add some points in that, but I think Mr. Sohail is also the right person to answer this. yes, the temperature change is one of the cause of this diseases. But the manufacturing of the malaria vaccine doesn't consider the climate change. It do consider the physiological parameters or the geographical locations, not the temperature, but the physiological conditions of the host or the patients, or the genetic diversity of the patients where the vaccine will be administered. So in broad sense, most of the malaria vaccine trials are being held in African countries. I mean, I don't know the exact reason. Maybe the reason is that malaria is more endemic there. But the irony is that the vaccines are being developed in developed countries, tested on several parameters, on mouse models of the in vitro stuff. So these are not the generic vaccines, but they don't consider the temperature. But temperature is a factor for vaccine storage. That was the issue for COVID vaccine, for the storage, right? But for the malaria vaccine, now at least they have this storage issue solved. It's a very good question, because for the Ebola vaccine, why things were taking long time? I'm adding Ebola because the vaccine which some companies developed in 2016-17, they had storage issue. So the transportation was a problem, because in endemic regions, you cannot carry -20 or the other high-end storage systems. So you can carry 4-degree storage systems. So now the Ebola vaccine has ability to be stored in 4 degrees centigrade. So they do consider this factor. So I will ask Mr. Sohail also to put some light on this. Thank you, Sohail.
Okay. Can maybe Mr. Sohail can add some an answer for this?
Yeah, thank you. Yeah, maybe I'm not the vaccine expert, but as I lived in the Indonesia, so and my city is also in the dengue actually. So I know the severity of the dengue and the malaria in Indonesia. But it seems about the environment. It's to me it's about the environment, and I agree with the Tarun about the vaccine actually is about some certain criteria to maintain that vaccine. I think the manufacture about the vaccine and the maintain the temperature, I don't have the good idea about it. But the one of the reason might be because Indonesia has some hilly areas actually. In Bangladesh actually, the malaria is not all over in Bangladesh, only in the hill tracts.
Yeah.
So in Indonesia basically, it's most happen in the hilly areas. So if you in the plain land, it is too hot. But if you go up to the mountain, it's too cold actually. So this difference of the environment or the temperature may impact about these two diseases actually, the malaria and the dengue.
Yeah.
It may happen.
I can add one thing in this, Sohail, that because the malaria vaccine will be given to people, it's not mandatory that they have to live in a tropical area, because they can travel to tropical area and
Right.
they go back to their own country, right, or own cities. So the vaccine will not be made considering the temperature of that area, because the vaccine should be able to work on all temperatures, right? Because changes in temperature is one of the cause, but imagine a person from Alaska is traveling to India and getting infected with malaria. So the vaccine should work on him also, right? Or they.
Yeah, this is very important. Basically, and also transportation
Yeah.
Transportation as well as the local transportation. Maybe I am receiving the vaccine in Bangladesh from the US to in Bangladesh, maybe in the good condition, but from Bangladeshi airport to the storage, from storage to the subdivisional level or the remote level, that storage may not maintain the proper SOP. So that may happen another cause actually. So that may the multiple reason may behind about this. Thank you.
Yeah.
Okay, thank you very much for the explanation for the board of speakers. I think we move to the next question. This is from Mrs. Dian. And this is to all speakers. As we all know, during the COVID-19 pandemic, the countries from all over the world have difficulty dealing with infection, as evidenced by the number of policies implemented to reduce the number of positive cases. So the question is, how far has the world been prepared to deal with the new infections and re-emerging so that when it happened, once again, the countries are better prepared to deal with this new, emerging and re-emerging disease? What should a country prepare to face this? I think we should go to the Mr. Tarun first.
Okay. So thank you for this question. So the simple answer for this question is that we don't know, because we don't know what will be the genetic diversity of the new infection, or whether it's viral infection, parasitic infections, or other infections. But the strong point is that we are technologically more advanced now. We were advanced before also, but now we have the tools that how to deal with the outbreak. First is that we have a good international connections, networks, and the organizations which work in tandem. We have media is always there to give you the updates, but the collaborations are more rigid now. The final thing which I want to emphasize is that virus, parasites, and other infection research are already there. They were there before COVID also. One thing that will change is the global travel, and also the international movement of students, which we are seeing in the US also, because the global travel, before we were planning to explore different wild forests. In India also, we traveled several forests. In USA, people now will be more aware, because the medical system with this global traveling has been updated after COVID. So if you are planning to travel somewhere, you get first all the checkups and all, and when you travel back to your country, then also you do that pre-screen, post screening. So that will stop things, but we don't know what is the source. Source is very important also, especially now these days, the food we eat, we sanitize our hands more. I'm sure before COVID, people were hardly sanitizing their hands when they were traveling in metros, buses, we were not thinking. But good thing is that being from Asia, we are immune to lot of stuff compared to developed countries. Yeah, that was a nice question.
Okay, Mr. Tran. I think that once again, the travelers is the biggest impact of why the infection disease happen all over the world, right?
Yeah.
Okay.
Can I add one thing? But
yes.
Also there is one reason of this emergence is the deforestation.
Okay. Yes.
Because you impact the ecology of this system, and new virus or new parasites evolve. So that is also important factor.
Okay. Well, okay. Thank you, Mr. Tyrone. To Mr. Sohel, please.
Yeah. Thank you. So, in addition to the Torun, actually, so Torun already says the many things actually. So actually, we have no controlling about this the infectious disease actually. But so what is my realization before COVID coming? So we are not aware about the IPC, infection prevention control. Actually, this infection prevention control mostly based on the hospital and the other health facilities, not in the community level actually, not in the much wider level. So this COVID actually teach us about this infection prevention control. Now we have the sanitize the hand regularly, hand washings, and the distantly, wearing the masks. So actually, this is the one thing for controlling. And the other thing about what I mentioned, the One Health. So this is not about the human being only. So it's also related to the animals and the environment as well. So we need to take care about our animals as well, the environment as well, to protect the re-emerging of the infection disease actually. So we stop that one. But we don't know when it will come, how it will come, but we don't know. But at least as a prevention or some prevention method we can do like this actually. Thank you.
Okay, thank you, Mr. Soil. And I think that this is the last questions. This is for me actually. I was wondering that will eventually, in your opinion, Mr. Tarun, will the society finally get herd immunity against malaria, considering that malaria is caused by virus, just like COVID-19? What do you think, Fathers?
So malaria is caused by protozoa, and herd immunity for malaria, no, it's not possible. Why? There are a couple of reasons. Because malaria infected mosquito bites a lot of people, but not everyone gets malaria. Couple of people gets malaria. So maybe, I mean, it's very selective, because there are some genes which attracts that, and some people are prone to malaria, some people are not. Second, some people by itself, they were not aware of, but they got immunity because they were infected with malaria when they were child, or when they were in school or something. So they already have the immune response or the antibodies, protective antibodies. And the third is that if there is outbreak of malaria, then that will be very severe. Then maybe we can think of the herd immunity, but I don't see an outbreak of malaria, at least in Asian countries, because there are some drugs already for malaria. And also with fever of 104.5, people understand, doctors are well aware, because from Calcutta, the first discovery started, of the malaria mosquito. And there will be no herd immunity stuff with malaria, I mean, for sure. Yeah. I don't know if some virulence. So there might be one problem if there is a recent research, I forget the lab in US, they are working on artificial mosquitoes or something, some genetically modified mosquitoes. So that is beneficial, but if they turn wild, if another mutation happens, then that can be more virulent also, you don't know. So yes, if the lethality increases, like for the infection, the falciparum, if there is some mutation and there are more deaths compared to the current scenario, then that will be very concerning, because now at least 80 to 90% people get well after getting infected with malaria. But if that percentage decreases, then that will be a big issue.
Okay, thank you, Mr. Tarun. Because I think that malaria in Indonesia, in some areas in Indonesia, still have high malaria cases, because
so
it's related,
vivax, right?
Yes, because it's related to their culture.
Yeah, it's mostly in the eastern area, right? The Papua,
Yes.
that area.
Yes, of course, that area.
Yeah, but that is not falciparum, it's Plasmodium vivax, right? Which don't cause cerebral malaria.
Yeah.
Right.
Yes.
So yeah, I know a couple of labs who have cohorts there. They get human samples from Singapore also, I think, and from USA, Florida also, they have some centers there. Yeah, I mean, they have to change some local administration. Local administration has to do something, you cannot help if the local administration sanitation is not important.
Yes. Thank you. Okay, thank you very much, Mr. Tarun. And apparently, we still have another question to be discussed, but due to the limited time, we need to end our discussion. What a very very knowledgeable of discussion today. So ladies and gentlemen, finally we have come to at the end of this part symposium. Before I close the session, to Mr. Tarun and Mr. Sohel, are there any final thoughts before we end today's discussion? To Mr. Tarun first.
Yeah, I mean, I will say thank you to all of you, all the eminent professors there and all the organizers. I know this was very hard work for a couple of days or weeks. You guys work very hard. The webinar went very smooth. I mean, last year when the webinars thing started, there were a lot of mishaps. So this was very nice, well organized, and the theme was very well thoughtful. So congratulations to all of you for organizing this, and thank you for invitation.
Thank you very much for Mr. Tarun. To Mr. Sohel, please.
Yes, yeah. Thank you for organizing this such a good webinar and the good topics. And terima kasih Ibu Istikes, Ibu Ketua Istikes, terima kasih Mbak Febri, and also thank you for inviting me as a speaker, and and thank you, and I'm delighted to join with you. And as I already have the Indonesian culture in my mind as I also there, so it's very happy for me actually to join any Indonesian event. So I used to join the other university, other polytechnic institute sessions actually. So it's for this time, so it's happy to join with you, and hope to work with you in future again. Thank you.
Thank you. Terima kasih Bapak Sohel. Okay. Finally, ladies and gentlemen, thank you for the great presentation. And finally, please allow me to take some highlighted points from this today's session. That once again, that emerging and re-emerging disease still a problem in some countries, maybe for all over the world. But one thing we should know that we need to do a collaboration between scientists, researcher, and any multidisciplinary, and how to handling these issues together. Okay. And then finally, ladies and gentlemen, I would like to thanks to the speaker, Mr. Tarun and Mr. Sohel, for their informative and interesting presentation, and also to the all attendees for their active participation. Thank you very much. So see you all later, and thank you very much. Over to you, Bu Fatmi. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Well, thank you, Ms. Febri Handini. We also thanks to our speaker, Mr. Tarun Kaswani and Mr. Sohel, who have delivered and beneficial material about emerging and re-emerging infectious disease, future challenges, and strategies. Thank you so much, sir.
Thank you so much.
Well, ladies and gentlemen, first Alifah Health Science Symposium from STIKES Alifah Padang comes to an end. We would like to thank you for your participation in today's webinar. Hopefully, this event will also be available place to share all the knowledge and experience for all of us. Okay, ladies and gentlemen, that's all for today. Let us close the webinar by saying Hamdalah together. Alhamdulillahirabbil alamin. We would like to apologize for any errors and shortcomings during the event. See you at the next webinar series from STIKES Alifah Padang next year, insyaAllah. Have a good day, everyone. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Mr. Tarun, Mr. Rohel.
Have a nice weekend.
Thank you for all.
Bye-bye.